Hi everyone, welcome back to Bio Explained. So I'm going to be running you through the entire GCSE biology course. I am going to be focusing on the AQA specification. If that doesn't apply to you, don't worry, GCSE biology is very similar between boards. So let's just get started with our first topic, which is cell structure. Okay, first things first. We've got two different types of cells. We've got prokaryotes and we've got eukaryotes. Now, eukaryotes are usually bigger, they're more complicated, and their genetic information, their DNA, is found inside a sophisticated structure called the nucleus. Prokaryotes, on the other hand, are much simpler. They are found in bacteria cells. They've got this weird cell wall structure, and instead of their DNA being found in sophisticated nucleus structures like in eukaryotic cells, instead they are found in single DNA loops or single small rings of DNA called plasmids, and they're just floating around in the cytoplasm of the prokaryotic cell. Just to clarify, the cytoplasm is found in eukaryotic cells and prokaryotic cells and is the jelly-like structure that makes up the body of the cell. It's where many chemical reactions take place. So what is an example of a prokaryotic cell? Bacteria. And what's an example of a eukaryotic cell? The very famous animal and plant cells. Let's go into a bit more detail about the substructures that make up these cells. Okay, starting with animal cells. They have the nucleus that contains the DNA or the genetic information. They've got a thin cell membrane that goes around the outside that allows substances in and out. And they've also got these small structures that are called mitochondria that make energy. They are the site for respiration, which is a process that makes energy. They're also known as the powerhouse of the cell, which is a very common phrase. Okay, now plant cells are more complicated, as you can see. They have some additional structures. They've got a cell wall going around the outside that is much thicker, and it's made of cellulose, which strengthens the cell. A plant cell does not want to be drooping with gravity, so it needs to be held upright, and the cellulose cell wall helps with that. They've also got these green structures called chloroplasts. They're green because they have the pigment called chlorophyll, which traps sunlight and allows for the process called photosynthesis, which allows the plant cell to make its own food. Animal cells don't need to make their own food. Plant cells do. And plant cells also have a permanent vacuole, as you can see here, which is filled with cell sap and gives the plant an additional source of sugar if it needs any. Okay, let's talk a little bit about specialization and differentiation. So, cells can be specialized. Animal cells, for example, can become specialized animal cells, such as sperm cells, nerve cells, or muscle cells. And root hair cells, xylem and phloem cells are examples of plant cells. Root hair cells are very specialized as the hairs increase the surface area, allowing for more water absorption in the soil, which is very useful for plants. And xylem and phloem have their own adaptations. Now, differentiation is very important. Cell differentiation in animal cells happens at an early stage, and in plant cells, even older plant cells have the ability to differentiate through their life. Differentiation is basically when a non-specialized cell becomes specialized. And when it's specialized, they have certain structures that allow it to carry out certain functions, so it makes it more useful. Let's look a little bit into the important skill of microscopy, which is using a microscope. So in biology, we need to know about two main types of microscope, the light microscope and the electron microscope. The light microscope is the one you will use in schools, and electron microscopes are much more complicated and usually require a skilled person to be able to use them. So why do we use each one? A light microscope is used to see the larger picture, and the electron microscope is used to see the smaller, finer details such as subcellular structures like mitochondria or chloroplasts. This is because the electron microscope has a much higher magnification and a much higher resolution. Both these things allow it to be used to see smaller structures in finer detail, making it much more powerful than the light microscope. Now let's look here at magnification and calculations. So this is the equation that you will be using for all your calculations, and there will be a calculation question that comes up as they're very common. You need to know this formula, which is image size equals to actual size times magnification. You can rearrange this formula for whatever you need to work out. So for example, in this example that I've written over here, if the image size is 10 millimeters over here and the actual size is 0.1 millimeters, okay, how do we find the magnification? So to use this triangle, you need to cover up what we're trying to find. So we're trying to find the magnification. Hence, the formula we need is I divided by A. So I'm going to write that out. I divided by A, or image size divided by actual size. So that's 10 millimeters divided by 0.1 millimeters. And the answer is, our answer is 100. And because it's asking for magnification, you need to put a little times before it. So the magnification is times 100, and that's your answer. I hope that makes sense. If you've got any questions, leave them down below, and thank you very much for watching.